My name is Yinqing. I'm a graduate student in Fengzhang's lab in the McGovern Institute for Brain Research and Broad Institute. In neuroscience research, we're kind of navigating without a map. Right now, the system neuroscience probe the brain by means of electrical recording, local manipulation. It's pretty much like you take a probe, you go into an electric circuit without the circuit diagram, and you probe like point by point, and you try to figure out how it works. That's really difficult. But once you have the connectome, it's like you have the circuit diagram. So connectome is about the connectivity of neurons and how they are wired up together. People has a long-term interest in connectome since 1970s. That's a time when people apply electron microscope techniques to study worm nerve systems. It took almost 10 years to be able to finish that. It's only involved 300 neurons. And when we go up to mammalian brains, let's say mouse, in a millimeter cubic, there are thousands of neurons and maybe billions of synapses connections. And how are we figure out their wiring diagram? It's going to take us years and years to finish. So my research involves a new approach to tackle the connectome problem, that is by sequencing. Right now, people can do sequencing billions of nucleotides in a few hours, actually. So if we can convert the connectome problem to a problem of sequencing, then we can solve that problem in a much timely manner and a cheaper way. My research in Fengzhang's lab involves primarily three parts. So the first part is to be able to individually label neurons with a barcode. What we call barcode is a unique nucleotide sequences. And so once we label individual neurons, then we have to associate their connection with those barcodes. And that's what we call the labeling the connections or synapses. And the third part of research is to be able to retrieve those connected associated barcodes that represent the synaptic connection. And at the same time, we can look at what are the neuronal identity genes being associated with that particular barcode. We'll be able to tell what kind of neuron type we're looking at. My career goal is to really solve the connectome. This may take decades, but I'm really interested in this. It will revolutionize potentially the way that system neuroscience asks questions and cure neuron diseases.